Hello students, today we will discuss on the topic ovule culture and embryo culture. So what is ovule culture? Ovule culture is a method by which ovules are aseptically isolated from the ovary and are grown aseptically on chemically defined nutrient medium under controlled conditions. The main objective of this ovule culture is to know the behavior of the zygote or very young embryo that are difficult to excise and also to culture them. In 1932, White first attempted to isolate and culture the ovule of a plant under aseptic conditions. Later on in 1958, Maheshwari perfected the technique of ovule culture through a series of experiments on different nutrient medium. Now, what is the principle of ovary culture? An ovule is a megasporangium covered by integument. Ovules are attached with placenta inside the ovary by means of its funicles. An ovule contains a megaspore or an egg cell. After fertilization, a single cell zygote is formed, which ultimately lead to form a mature embryo possessing shoot and root primordia. Ovule can be isolated and cultured in nutrient medium. In vitro ovule culture help to understand the factors that regulate the development of a zygote through organized stages to a mature embryo. Alternatively, it may be possible to germinate pollen in the same culture as the excised ovule and to induce in vitro fertilization and subsequently embryo production. To raise mature seeds by culturing, the ovules at the globular or later stage of the embryo development has been assayed in many species. Maheshwari in 1958 and in 1961 Nirmala Maheshwari and Lal could raise viable seeds of papaver from the ovules excised 6 days after pollination when they contain only a two-cell pro-embryo and a free nuclear endosperm. Seeds formed in the culture in 20 days and they germinated and produced seedling in the culture tube. In 1959 and 60, Prodymka Arnoldi succeeded in rearing ovule from pollinated ovary of some orchids on simply 10% sucrose solution. In 1959, Kapoor successfully grew the ovule of a plant at the zygote stage to mature seeds, and such seeds often germinated in situ. It is to be mentioned that in many plants, mature seed could be obtained from the ovule culture at the zygote or two-cell embryo stage only, if the medium was supplemented with coconut milk and fruit juice. The ovules of Gossypium hirsutum were cultured by Joshi and Johri in 1972 on a medium supplemented by casein hydrolysate. In all cases, tannin was accumulated in the cotton fiber initials and as such the initial collapsed. However, Beardsley and Tim in 1973 have shown that the better development of fibers takes place at 30 degrees centigrade if excised ovules are cultured 2 days after anthesis on a medium supplemented by gibberellins.